Hello everyone, I'm Dr. Arpita. A very warm welcome to all of you here. Today we are going to discuss about glycosylation of proteins. Glycosylation of proteins is a ubiquitous modification, okay, of newly synthesized proteins in the endoplasmic reticulum. It is dependent on the linkage of oligosaccharides or sugar moieties, or simply sugars you can say, to the amino acid side chain of the protein. There are two major types of glycosylation which we will discuss soon. One is the N-linked glycosylation and the other is O-linked glycosylation. There are other few types of glycosylation, but you need not know the details of that. But here we are going to discuss N and O type or N and O linked glycosylation. But just for the sake of your knowledge, I'm telling you glycosylation and its types. Okay. First, N-linked glycosylation. What happens here? Glycan binds to the amino group of asparagine in the ER. In the endoplasmic reticulum. Okay. Next, O-linked glycosylation. Here what happens? Monosaccharides bind to the hydroxyl group that is the OH group of serine or threonine in the ER, then Golgi, cytosol, and nucleus. C-linked glycosylation. What happens here? See, mannose binds to the indole ring of tryptophan. Mannose binds to the indole ring of tryptophan. And then what do we have? Phosphoglycosylation. What is happening here? The glycan binds to serine via phosphodiester bond. So these are the four major types of glycosylation, but we are only concerned with these two. So glycosylation is a critical function of the biosynthetic secretory pathway in the ER and Golgi apparatus. Keep in mind, approximately half of all proteins typically expressed in a cell undergo this modification, which entails the covalent addition of sugar moieties to specific amino acids. Now, we should all know what are the various advantages of peptide glycosylation. The first one, targeting specific organs and enhancing biodistribution in tissues. So this is very important for the purpose of targeting. Okay. Improving penetration through biological membranes. This modification helps in penetrating through biological membranes, which are extremely selective. Next, increasing metabolic stability and lowering clearance rate. It helps to increase the metabolic stability and thus help in lowering the clearance rate. Then it helps in receptor binding, and finally it protects amino acids side chain, which is very important. These diagrams, you can look into it later. First, let us discuss what is happening in the event of glycosylation. Okay. Protein N-glycosylation is a metabolic process. As we have just discussed, it is a metabolic process that has been highly conserved in evolution. In all eukaryotes, N-glycosylation is obligatory for viability. You cannot bypass this. Its functions, it functions by modifying appropriate asparagine residues of proteins with oligosaccharide structures, thus influencing their properties and bioactivities. So the amide nitrogen of amino acid is the point of attachment of oligosaccharide. That is the sugar moiety. So the amide nitrogen of the amino acid for N-linked glycosylation, this amino acid is asparagine. Okay, this is asparagine. The amide nitrogen of the amino acid is the point of attachment of oligosaccharide, that is the sugar moiety, and the name of the bond formed here is N-linked glycosidic bond. So this is the bond that is forming here. See, you must keep in mind what is the main event that is happening and which is the amino acid that is associated with which type of glycosylation. Because it is always transferred to the amide nitrogen or like NH2 group on the side chain of asparagine residue of the protein, this oligosaccharide is said to be N-linked or asparagine linked. Okay, this transfer is catalyzed by a membrane-bound enzyme. This is an important point. This transfer is catalyzed by a membrane-bound enzyme known as glycosyl transferase. With its active site exposed to the luminal surface of ER membrane. Exposed to the ER membrane. Clear? The preformed precursor, keep in mind the preformed precursor is transferred en bloc to the target asparagine residue in a single enzymatic step almost as soon as that residue emerges in the lumen of ER during protein translocation. And since most proteins are co-translationally imported into the ER, N-linked oligosaccharides are almost always added during protein synthesis and ensuring maximum access to the target asparagine residues which are present in the sequence. The sequence you should know, which are present in the sequence or excuse me. Excuse me. So here X is the amino acid except proline. X is the amino acid except proline here. So these two sequences please pardon me for my bad throat today. So these two sequences thus function as signals for N-linked glycosylation. Now come to the diagram here. See, as I told you, the non-glycoproteins formed on the ribosomes move into the lumen of ER cisternae immediately after polypeptide chain synthesis. Okay. Now, proteins destined for glycosylation, they remain attached to the ER membrane, which contain glycosyl transferases that catalyze glycosylation of protein. See, we have discussed it here just a while ago, this enzyme. Where did we write that? Here, if you take a look, we have mentioned about this enzyme. Now, the first sugars may be attached to the polypeptide chain in the ER cisternae, even while the chain is attached to the ribosome. So this concept has to be there instilled in your mind. Now come to the diagram. In step 1, what you see here, the growing protein translocates across the ER membrane. Good enough. Now this dolicol is the lipid carrier here. Coming to the next step. What's happening? You see asparagine X, which is an amino acid, and here serine or threonine. We have just talked about these. Now see here. Oligosaccharide transferase transfer oligosaccharide from dolicol to asparagine. This was the carrier, isn't it? So from here, the oligosaccharide is transferred to asparagine. Who is doing this job? Who is doing this job? Oligosaccharide transferase. Fine. So it is transferring the oligosaccharide from dolicol to asparagine. Here, once the work is over, the enzyme is set free. This enzyme, 2 enzymes remove 3 sugars from oligosaccharide. Clear? Step is very simple here. This is a simplified diagram. If you look into this, this is also giving the same message, but a little elaborate diagram. Okay, so this is all about N and O linked glycosylation. Hope you have understood the concept and have enjoyed learning. Then, thank you for your attention. We'll meet soon with a new topic. Till then, goodbye.